Welcome to the deep dive. Today, we're really getting into something fundamental, the intricate dance between DNA and, well, its dynamic partner, RNA.
Hmm.
We always hear DNA as the blueprint, but understanding RNA is just as vital for, well, for grasping everything from heredity to the latest medical stuff.
That's absolutely right. And for this deep dive, we've pulled together insights from some core cell biology sources and also broader genetics overviews.
Okay.
Our goal here, really, is to pull out the key nuggets of knowledge, help you understand their crucial differences, how they actually work together, and maybe why getting a handle on these molecules is kind of a shortcut to being well-informed about, well, the essence of life itself.
Yeah, I think there are going to be some real aha moments here. Some surprising facts, maybe, and definitely looking at the real-world applications that kind of hinge on these tiny, tiny molecular distinctions. All right, let's unpack this. So starting high level, it seems, well, it's easy to see DNA and RNA as a pair, isn't it? They're both linear polymers, essentially these long chains built from repeating units.
Exactly.
Right.
Built from sugars, phosphates, and these nitrogenous bases.
Right.
They share those core building blocks, absolutely. But let's say their responsibilities in the cell are quite distinct, almost like different departments in a factory.
Okay.
DNA is really for the long-term storage of genetic info. RNA. That's more about reading, interpreting, and putting that information into action.
And those differences allow them to work together, underpinning all life.
Precisely. It's that teamwork based on their differences.
And when we dig into those differences, our sources seem to group them into 5 key areas that define their roles.
Yep, 5 main categories.
We're going to look at differences in function, the type of sugar, their bases, overall structure and length, and location in the cell. So let's kick off with function.
Okay, function. Think of DNA as the cell's master archive, like a biological flash drive, but super secure and long-term. It stores all the genetic information, the whole blueprint for life. And critically, it's built for stability,
Yeah.
to be passed down accurately between generations. That's key for continuity.
Okay, so DNA is the secure archive, then
Yeah.
RNA is like the active workforce reading it.
That's a great way to put it. Yeah, the dynamic workforce. And it's not just one type of RNA doing everything.
Ah, right. Our sources mentioned 3 main types.
3 really important ones involved in making proteins, which are the cell's workhorses.
Okay.
First up, you've got messenger RNA, mRNA, its job, to copy specific bits of the genetic code from the DNA. That process is called transcription.
Transcription, okay.
Then it transports those copies, like sending a memo, over to the cell's protein factories, the ribosomes.
Okay, so mRNA carries the specific instructions from the blueprint. How do those instructions then get, built into an actual protein? What else is involved?
Good question. That's where transfer RNA, or tRNA, steps in.
tRNA.
This molecule basically acts like a delivery truck. It brings the specific building blocks for proteins, those are called amino acids, to the ribosome factory.
Okay.
And it doesn't just bring any amino acid, it matches them precisely to the coded instructions brought by the mRNA. That whole building process, that's called translation.
Translation, got it. Transcription is DNA to mRNA. Translation is mRNA to protein. And the third RNA type, you said it was foundational.
Absolutely foundational. That's ribosomal RNA, or rRNA.
RNA.
Now, this one isn't just carrying messages or parts. It's actually a core structural and catalytic part of the ribosome factory itself.
Oh, interesting. So it's part of the machinery.
Exactly. Without RNA, the ribosomes, the protein factories just wouldn't work, period.
Wow. Okay, so pulling that all together. DNA holds the master plan, mRNA copies a specific instruction, tRNA brings the parts, and RNA is part of the factory that builds the protein.
You got it.
And this whole flow, DNA to RNA to protein, that's the central dogma of molecular biology.
All right. That's the one. The core information pathway. Pretty much universal in life.
Okay, let's move to the next key difference. The sugar molecule in their backbones.
Right, the sugar. Both have a sugar phosphate backbone, but the sugar itself is slightly different. DNA has deoxyribose.
Deoxyribose?
RNA has ribose. And that deoxy prefix in DNA is sugar. That's the clue. It means deoxyribose has one less hydroxyl group. That's an oxygen and a hydrogen, OH, compared to ribose.
And that one little chemical difference makes a huge difference functionally, doesn't it?
It really does. RNA's extra OH group makes it more reactive, sort of more flexible chemically, which is actually useful for short-term jobs in making proteins.
Okay, more reactive. And DNA.
DNA, with that missing oxygen in its deoxyribose sugar, is much more stable chemically. less reactive.
Which makes perfect sense for something that needs to store precious genetic information safely for, well, potentially a lifetime.
Exactly. Stability is paramount for the archive.
Okay, third difference. The nitrogenous bases. The letters of the code.
Right, the bases. In DNA, you have 4: adenine, thymine, guanine, and cytosine. Usually just called A, T, G, and C.
ATGC.
And they pair up very specifically. A always pairs with TC, always pairs with G.
ATCG.
Got it. Now, RNA, this is where it gets interesting. RNA uses A, G, and C, just like DNA. But instead of thymine, T, it uses a different base called uracil, U.
Uracil, so RNA has U instead of T.
Correct. So in RNA, the pairings you see are A with U and C with G.
AUCG. Why the difference? Why uracil?
Well, it's related to that stability point again. Partly, thymine is slightly more stable, better for long-term DNA. Uracil is maybe a bit easier to produce and use for the temporary RNA copies.
Yeah.
It also helps the cell clearly distinguish between DNA and RNA instructions.
Makes sense. Okay, fourth difference, structure and length.
Structure and length, DNA is the famous one, right? That double helix.
The twisted ladder.
Exactly. 2 long strands of nucleotides wound around each other. And these molecules are long. A single human chromosome, which is one DNA molecule, can be centimeters long if you stretch it out.
Centimeters. Wow. Okay, and RNA.
RNA is generally much simpler, usually just a single strand.
Single-stranded.
And much, much shorter than DNA.
Yeah.
Maybe only a few thousand base pairs long compared to millions or billions in DNA.
So shorter, single-stranded. Any exceptions?
Well, our sources do mention that RNA can fold back on itself sometimes and form temporary short double helix sections. It's not its main state like DNA, but it adds to its versatility.
Interesting. Okay, final key difference. Location. Where do we find them in the cell?
Location reflects function.
Right.
Again, the vast majority of DNA, at least in eukaryotes like us animals, plants.
Right, cells with a nucleus.
Exactly. Most DNA lives inside the nucleus. It's packaged really tightly into those structures called chromosomes, keeps it safe and organized.
Makes sense for the master archive. Any DNA elsewhere?
Yep. A small amount is also found in the mitochondria, the cell's powerhouses. They have their own little bit of DNA.
Okay. And RNA. It's the workforce, so it's probably moving around more.
Totally. RNA's journey is much more dynamic. mRNA, the messenger, it's actually made in the nucleus, copied from the DNA template there.
Okay.
But then it travels out of the nucleus into the main body of the cell, the cytoplasm. Gets transported around by the cell's internal skeleton, the cytoskeleton.
So it leaves the nucleus. What about tRNA and rRNA?
tRNA, the transfer RNA that carries amino acids, that just roams freely in the cytoplasm, ready to grab its specific amino acid when needed.
Okay.
And rRNA, the ribosomal RNA, well, ribosomes themselves are actually assembled in a special part of the nucleus called the nucleolus.
Ah, the nucleolus.
Right. Then the finished ribosomes, containing RNA, are exported out into the cytoplasm to start building proteins. So RNA is made in the nucleus, but does most of its work out in the cytoplasm.
That paints a really clear picture. The 5 key differences really highlight their specialized jobs.
Absolutely. Function, sugar, bases, structure, location. They all tie back to DNA being the stable archive and RNA being the dynamic worker.
Now, you mentioned stability earlier because of the sugar difference, deoxyribose making DNA more stable.
Yes, that lack of an oxygen atom in the sugar really cuts down on its reactivity compared to RNA's ribose. It's much better suited for keeping that genetic information intact over long periods. RNA's reactivity is actually good for its short-term roles. Easy come, easy go, basically. It can be made quickly and then broken down when the job's done.
And what about things like UV light? Does that affect them differently?
It does. DNA is actually quite vulnerable to damage from UV radiation. That's why sun exposure can cause skin cancer mutations. RNA, interestingly, is generally more resistant to UV damage.
Okay, so these core differences are clear, but you also hinted that it's not always just the standard BDNA, double helix, and single-stranded RNA. There are unusual types.
Oh yes, life is incredibly diverse, right down to the molecular level. Beyond the standard forms, there are some really fascinating variations.
Like what?
Well, for instance, there's something called zDNA.
zDNA.
Yeah. Most DNA, the kind Watson and Crick described, is a right-handed helix. We call it bDNA. zDNA is a left-handed helix.
Left-handed. Wow.
It's thinner, has a different structure. It was quite a surprise when discovered. It seems to form temporarily in certain situations, perhaps behind enzymes moving along the DNA, and might play a role in regulating genes. It shows DNA isn't just a static structure.
Fascinating. What else?
There's also aDNA. This form often shows up when DNA is dehydrated, like you might find in bacterial spores.
Okay.
It's shorter, more compact than bDNA. For a long time, its role wasn't clear, but now we think it's important for how enzymes bind to DNA, maybe exposing certain parts for them to work on. And, like RNA, aDNA is much less susceptible to UV damage, so bacteria-forming spores might use it as protection.
Protection against harsh conditions. Makes sense. Any others?
Then you get into really complex structures like triplex DNA, sometimes called hDNA.
Triple helix, how does that work?
Yeah, a third strand winds around the standard double helix. It can happen naturally under certain conditions, or scientists can engineer short strands, called oligonucleotides, to bind to specific DNA sequences, forming a triple helix.
Why would you want to do that?
It can be used to target specific genes, maybe block their activity, or deliver modifying agents right to that spot. Interestingly, some natural forms like hDNA are actually linked to increased mutation rates in those regions. So DNA can be surprisingly dynamic, even mutagenic itself sometimes.
Wow. Okay, so DNA isn't always just the stable archive. And
Sure.
what about unusual RNA?
Well, the big one is double-stranded RNA or dsRNA.
Wait, I thought RNA was single-stranded.
Usually, yes. But dsRNA exists as a stable double helix. You find it most often as the entire genome of certain viruses.
Viruses using RNA as their main genetic material.
Yeah.
Double-stranded.
Yep. Lots of plant viruses. And some animal and human ones, too, like rotaviruses that cause severe diarrhea, especially in kids.
Okay.
But the really revolutionary thing about dsRNA is how our cells react to it. It acts as a powerful trigger for the immune system. It's like a big red flag saying virus.
Ah, so our bodies recognize it as foreign.
Exactly. And understanding how cells recognize and respond to dsRNA led directly to the discovery of a process called RNA interference, or RNAi.
RNAi, I've heard of that. Gene silencing.
Precisely.
Oh, yeah.
It's a natural mechanism cells use, partly triggered by dsRNA, to shut down specific genes, harnessing that mechanism. That led to RNAi technology, which won the Nobel Prize back in 2006. It's a major research tool and has huge therapeutic potential.
Incredible. From understanding a viral genome to a Nobel Prize-winning technology.
It shows how fundamental this stuff is.
Okay, let's zoom out a bit then. Connect these molecular details to the bigger picture of genetics. DNA and RNA are clearly the stars of the show.
Absolutely. They're the core players. Cells are the basic units of life, and DNA made of just those four bases, A, T, G, C, holds all the instructions. The sequence matters.
And the scale is just mind-boggling. The human genome, something like 20,000 to 25,000 genes.
That's the estimate, yeah. But what's also wild is that genes themselves only make up about, maybe 29% of the entire genome?
Only 20%. What's the rest?
A lot of it is non-coding DNA. Some provide structure to the chromosomes. Some is involved in regulating when and where genes get turned on or off. It's definitely not all junk DNA, as people used to think. It has crucial roles.
And even though, say, my liver cell and my brain cell have the exact same set of genes, the same DNA.
Right, the full complement. Humans have 46 chromosomes, 23 pairs in almost every cell.
They use different parts of that blueprint, don't they?
Exactly. Selective gene use. A liver cell needs to make liver enzymes, so it turns on those genes. A brain cell needs neurotransmitters, so it uses a different set of genes. It's all about expressing the right genes at the right time. Or, like you said, environmental triggers like an infection can cause certain genes to switch on.
Which brings us right to that classic nature versus nurture debate, doesn't it?
It really does.
Yeah.
Because the phenotype, the observable traits you see in an organism,
Like eye color, height, that kind of thing.
yeah, or even susceptibility to disease.
Yeah.
That phenotype depends on the interaction between the genes, the nature part, and the environment, the nurture part.
Can you give an example?
Sure. A classic one is the coat color in Siamese cats.
Ah, with the darker ears and paws.
Exactly. They have a gene for dark fur pigment, but the enzyme that makes the pigment is temperature sensitive. It only works properly in cooler temperatures.
So the cooler parts of their body, ears, paws, tail, get the dark fur.
Right. And the warmer torso stays lighter. Same gene everywhere, but the environment temperature changes how it's expressed visibly.
That's a great visual. What about a human example?
Phenylketonuria, PKU, is a powerful one. It's a genetic disease. A mutation means you can't break down an amino acid called phenylalanine.
Okay.
If it builds up, it causes severe problems, intellectual disability, seizures. But if you know a baby has the PKU gene and you put them on a strict diet from birth that avoids phenylalanine,
They can be okay.
they can develop completely normally. The environmental intervention, the diet, completely changes the outcome of the genetic predisposition. It's a dramatic example of gene-environment interaction.
Wow. And scientists study this interaction using twins, right?
Yeah, twin studies are crucial. Comparing identical twins who share 100% of their DNA with fraternal twins who share about 50%,
Hmm.
like any siblings. If identical twins are much more likely to both have a trait or disease compared to fraternal twins, it suggests a strong genetic component.
Any famous cases?
The Gename quadruplets are a well-known, though complex, case study. They were identical quadruplets, all four of whom developed schizophrenia, which provided insights into the genetic underpinnings of the disorder, although environment clearly played a role in the variation of their conditions too.
So we all have variations in our DNA. You mentioned we're 99.9% alike, but that 0.1% difference accounts for, well, everything that makes us unique individual.
Pretty much. That tiny fraction explains differences in physical traits, disease risks, how we
Mhm.
respond to medicines. It comes in a couple of main flavors, though.
Okay.
We often talk about mutations. These are changes in the DNA sequence, and often they're linked to diseases. They tend to be relatively rare. It could be a single base change or deleting or adding pieces of DNA.
So mutations are often problematic. What's the other type?
Polymorphisms. These are also variations in the DNA sequence, but they're considered normal, natural variations. They don't usually cause adverse effects, and they're much more common in the population.
Can you give examples of polymorphisms?
Sure. SNPs, single nucleotide polymorphisms, are the most common. Just a single letter difference. You have millions of SNPs in your genome, making you unique. Another familiar example is blood types A, B, AB, O. Those are determined by common variations, polymorphisms, in a specific gene.
Okay, so mutations are rare, often harmful changes, while polymorphisms are common, usually neutral variations.
That's a good general distinction, yeah.
Mm.
And these variations contribute to different types of genetic diseases.
How so?
Well, you have single gene disorders, like PKU or cystic fibrosis, caused by a mutation in just one gene. Then there are chromosomal disorders.
Like Down syndrome.
Exactly. Down syndrome is usually caused by having an extra copy of chromosome 21. That's a type of aneuploidy, an abnormal number of chromosomes. Other chromosomal issues involve large deletions or rearrangements of chromosome structure.
Okay, single gene, chromosomal, what else?
And then there are multifactorial diseases. These are really complex. They result from a combination of variations in multiple genes interacting with environmental factors and lifestyle choices.
So things like heart disease, diabetes, maybe some birth defects like spina bifida?
Precisely. Those have genetic components, but your risk is also heavily influenced by diet, exercise, exposure to certain things. It's a complex interplay.
And underlying all of this passing down of traits and variations are the basic rules of inheritance, right? Mendel's laws.
Yes, Gregor Mendel laid the groundwork. Key ideas like traits being inherited as discrete units, not just blended together. The idea that gene versions or alleles segregate during reproduction, and that different genes are often inherited independendly of each other.
And geneticists use tools to track this, like those charts.
Punnett squares, yeah, to predict inheritance probabilities for simple traits, and pedigree charts to trace traits through family histories. Those are fundamental tools.
Okay, so we've covered the molecules, their differences, the broader genetics. Let's talk about the cutting edge. How is this fundamental knowledge being applied today?
Oh, it's everywhere. In medicine, DNA sequencing is now routine for diagnosing rare genetic disorders, sometimes even guiding cancer treatment by sequencing tumor DNA,
Right.
and in biotechnology. Manipulating DNA is the basis of genetic engineering. Think modified crops, bacteria engineered to produce insulin or other drugs. It's huge.
And then RNA is having a major moment too, isn't it? You mentioned RNAi.
Absolutely. RNA interference, born from understanding the dsRNA response, is a powerful tool to specifically turn off genes. Huge potential for treating diseases caused by faulty genes.
And of course, the RNA vaccines.
Yes, the COVID-19 mRNA vaccines were a massive breakthrough, bringing RNA technology right into the mainstream. They showed how quickly and effectively mRNA could be used to instruct our own cells to build immunity. It really highlighted RNA's dynamism.
It feels like RNA went from being DNA's assistant to a star player in its own right.
In many ways, yes. We're appreciating its versatility much more. And understanding genetics is also key for tackling things like cancer.
How so? You mentioned mutations earlier.
Cancer is fundamentally a disease of the genome. It arises from an accumulation of mutations in a cell's DNA somatic mutations, meaning they happen during life. They aren't usually inherited.
Okay.
These mutations might disable important break genes like the p53 tumor suppressor or jam the accelerator by activating genes like WAS proteins, leading to uncontrolled cell growth. Understanding these genetic changes is critical for developing targeted cancer therapies.
And all this progress relies on powerful research tools, right?
Yeah, definitely. We rely heavily on model organisms, fruit flies, worms, yeast, mice, even bacteria like E. coli. They let us study gene function and disease in simpler systems.
And techniques to work with the DNA itself.
Oh yeah. PCR polymerase chain reaction is indispensable. It lets scientists amplify tiny amounts of DNA, making it possible to study minute samples like in forensics or diagnosing infection.
Right, making lots of copies from a tiny starting amount.
Exactly. And then there's DNA sequencing technology. It's become incredibly fast and cheap compared to even a decade ago. Next-generation sequencing has fueled entire fields like genomics, studying whole genomes and bioinformatics, which uses computers to analyze all that data.
The Human Genome Project finishing in 2003 seems like ancient history now in terms of sequencing capability.
It really does. It laid the foundation, but the pace of change since then has been extraordinary.
But this incredible power to read and even edit DNA and RNA, that brings up some serious ethical questions, doesn't it?
Profound ones, absolutely. As our ability to understand and manipulate the building blocks of life grows, so does our responsibility.
I remember hearing about calls for a ban on certain types of gene editing.
Yes, particularly around inheritable human genome editing. Using tools like CRISPR or zinc fingers to make changes to sperm, eggs, or embryos, changes that would be passed down to all future generations.
Germline editing.
Right. There was a significant call from leading scientists back in 2015 for a moratorium on the clinical use of these techniques due to safety concerns and huge ethical implications. Research continues, sometimes on non-viable human embryos, to understand the possibilities and risks. But the debate about where to draw the line is intense and ongoing.
It really underscores why having a solid, impartial understanding of the science itself is so important for everyone, doesn't it? So we can participate meaningfully in these crucial discussions.
Couldn't agree more. Informed public debate is essential when the science touches on such fundamental aspects of who we are and what our future might look like.
Well, that feels like a good place to bring our deep dive toward a close today. We've gone from the nitty-gritty of deoxyribose versus ribose,
AT versus AU.
all the way to how these molecules define us, impact our health, drive evolution, and now even shape cutting-edge medicine and profound ethical debates.
It really shows that this knowledge isn't just, textbook stuff. It's actively transforming our world, changing medicine, and really challenging us to think hard about the future of life itself.
So the thought to leave everyone with: as scientists keep digging deeper, uncovering more unusual forms of DNA and RNA, figuring out even more complex functions.
Yeah.
What's next? What new frontiers in health or personalized medicine, or maybe even things we can't yet imagine, might open up?
And crucially, how will our evolving understanding of these fundamental building blocks shape the ethical decisions we'll inevitably face with tomorrow's scientific breakthroughs?
The dive, it seems, never truly ends. Thanks for listening to The Deep Dive.